Somewhere in your body right now, there is a small population of fat cells doing something almost no other fat cell does, burning through fuel as fast as it can get its hands on it. Most of what people call fat exists to do the opposite, to hoard energy, to expand quietly, to wait. This other kind works like it's being chased. It pulls in glucose and fatty acids and converts them almost immediately into heat, with nothing stored, nothing saved for later. For most of the 20th century, this fat type was treated as a footnote in human biology. Textbooks placed it firmly in the category of newborns and hibernating animals, useful for a baby that cannot yet shiver, or a rodent surviving a long winter, but irrelevant once a person reached adulthood. That assumption held for decades, repeated in medical education without much challenge, until a wave of imaging studies quietly dismantled it. What those studies found, and why it took so long to notice something sitting in plain sight inside adult bodies, is something we'll get into later in this video, along with why some people appear to carry far more of it than others. Quick thing before we go further. If you're watching this because you actually want to do something with your metabolism, not just understand it, I put together a simple daily system based on the same kind of research we're covering here. It's linked in the first pinned comment if you want to check it out. Now, back to where this fat actually comes from. What actually separates this energy-burning fat from ordinary fat comes down to three things: where the cells come from during development, what's packed inside them at a microscopic level, and a single protein that does something no other fat cell protein does. Understanding those three pieces is really understanding the whole story. So that's where we start. Picture a typical fat cell, the kind that makes up the bulk of body fat in every adult. Under a microscope, it's almost embarrassingly simple. One enormous droplet of stored lipid fills nearly the entire cell, pushing the nucleus and everything else to a thin rim along the edge. There are very few mitochondria inside, because this cell isn't in the business of burning fuel. Its job is storage, plain and simple. A long-term energy reserve the body can draw on when food is scarce. Researchers studying adipose tissue structure, including work published by Saverio Cinti in the Journal of Endocrinological Investigation in 2002, describe this arrangement as unilocular: one lipid pool, one droplet, one purpose. Brown fat looks almost nothing like this. Instead of a single droplet, the cell holds many small ones scattered through the cytoplasm, a multilocular pattern, and the space around them is packed with mitochondria so dense that they give the tissue its actual brown color under iron-rich cytochrome proteins. Cannon and Nedergaard's extensive review in Physiological Reviews, published in 2004, lays out how this isn't a cosmetic difference. Each of those mitochondria is preparing to do something white fat mitochondria essentially never do: convert stored fuel directly into heat rather than into cellular energy for later use. That structural gap reflects two completely different survival strategies built into the same broad category of tissue. White fat plays a long game, banking calories against future famine. Brown fat plays a short, urgent one, burning through fuel in real time, without banking a single calorie, the moment the body signals it needs warmth or a change in energy balance. Same starting material, same fatty acids and glucose circulating in the bloodstream, but two entirely different endings for where that fuel goes. Here's where the story gets genuinely strange. For years, scientists assumed that fat cells, all fat cells, regardless of color, came from a shared lineage of precursor cells, distinct from muscle, bone, or anything else. Patrick Seal and colleagues, publishing in Nature in 2008, overturned that assumption using a technique called in vivo fate mapping in mice, essentially tagging precursor cells early in development and tracking exactly what they became. What they found was that brown fat cells, unlike white fat cells, arise from precursors expressing a gene called MyF5, a gene that had previously been considered a marker exclusively for developing skeletal muscle. In other words, at some early developmental branch point, cells that could have become muscle fiber instead became brown fat, and the switch between those two fates turned out to hinge on a single regulatory protein called PRDM16. Remove PRDM16 from these precursor cells in mice, and they drift toward becoming muscle. Force PRDM16 into ordinary muscle precursor cells, and they convert into brown fat cells instead. It's worth being precise about what this tells us and what it doesn't. This fate mapping work was done in mice, using genetic tracing tools that can't ethically be applied the same way in human embryos, so scientists can't directly watch the equivalent switch happen in a developing human. But the shared genetic signature between brown fat and muscle, the mitochondrial density, the oxidative machinery, the underlying molecular toolkit, lines up closely enough across species that researchers treat this muscle-adjacent origin as the most likely explanation for why brown fat looks and behaves the way it does, even in humans. Every mitochondrion in your body runs on the same basic principle. Electrons move through a chain of proteins. That movement pumps protons across an inner membrane, and the resulting gradient drives an enzyme that manufactures ATP. The molecule cells spend as usable energy. This coupling of electron flow to ATP production is how the vast majority of your cells stay powered. Brown fat mitochondria break that rule on purpose. Sitting in the inner membrane of these mitochondria is a single protein called UCP1, short for uncoupling protein 1. French researcher Daniel Riquier, working with Jean-Claude Cotter, first identified this protein in rat brown fat mitochondria in 1976, publishing the finding in Biochemical and Biophysical Research Communications, with the protein's structure characterized more fully several years later. What UCP1 does is create a controlled leak in that proton gradient, a shortcut that lets protons flow back across the membrane without passing through the ATP-producing enzyme. The energy that would have built ATP is released instead as pure heat. The scale of this is worth pausing on. In active brown fat, UCP1 can account for a meaningful share of all the protein embedded in the mitochondrial membrane, according to the Cannon and Nedergaard review. Some researchers studying the evolutionary history of this protein have proposed that its emergence may have played a role in helping early mammals survive cold climates and the vulnerable window right after birth, though this remains an interpretation of the evolutionary record rather than something that can be directly proven. What's not in dispute is the mechanism itself, a single protein, quietly rerouting energy away from storage and into warmth every time it's switched on. Just when the white versus brown picture seems settled, there's a third category that complicates it. In 2012, Jun Wu and colleagues, working in Bruce Spiegelman's lab, published a study in the journal Cell describing cells they isolated from white fat adipose in mice. Cells that weren't classic brown fat and weren't ordinary white fat either. They called them beige adipocytes, and the name stuck. These beige cells don't come from the MyF5 positive muscle-adjacent lineage that produces classic brown fat. Instead, they emerge directly within white fat tissue, carrying a gene expression signature distinct from both of their neighbors. At rest, they behave almost like ordinary white fat cells, sitting quietly with low thermogenic activity. But under the right stimulus, they can switch on a thermogenic program almost identical to brown fats, recruiting UCP1, reorganizing their mitochondria, and starting to burn rather than store. Researchers call this process browning, and it turns out to be highly inducible, which becomes important later in this video. Here's the detail that reshaped how scientists think about adult human brown fat entirely. When Wu and colleagues examined human tissue samples in that same study, the deposits that had been identified in earlier adult imaging studies, the ones assumed to be classic brown fat, showed a gene signature that looked far closer to beige fat than to the classic brown fat found in mice. That finding blurred a line researchers had treated as fixed, and it raises an obvious question. If this fat can be switched on, what actually switches it on in a living adult body? That's less about temperature than most people assume, and the answer involves everything from a hormone released during exercise to a compound sitting in your spice rack. The image most people carry around of brown fat, something babies have and adults mostly don't, took a serious hit in 2009 when Aaron Cypess and colleagues at Harvard published findings in the New England Journal of Medicine that came from an unusual source. Nearly 4,000 PT-CT scans originally ordered for entirely different medical reasons, mostly cancer screening, which happened to also capture whether patients had detectable metabolically active brown fat in the neck-to-chest region. The results were reproduced almost simultaneously by two other independent research groups publishing in the same issue of the journal, which is part of why the finding landed with so much weight in the field. What the data showed was not a uniform picture at all. Brown fat activity showed up in just over 7% of women scanned, compared with just over 3% of men, more than double the rate, a gap researchers have since confirmed repeatedly. It also declined steadily with age, was less common in people using beta blocker medications, and, among older patients specifically, was far less likely to appear in those carrying a higher body mass index. None of these patterns tell you why brown fat behaves this way at a mechanistic level. They're associations pulled from a large data set, not an explanation, but together they paint a fairly consistent portrait. Younger, leaner, and female skews toward more of it. That raises an obvious question that these observational scans can't answer on their own. Is this fat type something you're simply born with more or less of, fixed for life? Or is it something that responds to what a body actually does and experiences? The honest answer, based on everything researchers have found since, is a mix of both. And the response to part turns out to be more interesting than most people expect, because the switches that turn this fat on have almost nothing to do with what most people assume. If anything so far has shifted how you think about your own body's fat, especially the fact that this has far less to do with willpower than most people assume. Tell me in the comments which detail surprised you the most. I read everyone, and it shapes what I dig into in future videos. That question, whether this is something you're just stuck with or something you can actually influence, is exactly the kind of thing I like turning into simple, practical steps instead of leaving it as theory. If that's useful to you, it's linked in the first pinned comment. Worth a look before we get into what actually flips the switch. Cold and capsaicin work through the nervous system, triggering a cascade of signals that eventually reach thermogenic fat cells from the outside. Thyroid hormone works differently. It's already circulating in the bloodstream, and thermogenic fat cells have the machinery to convert it locally into its most active form. An enzyme called DIO2 sits inside these cells and converts circulating T4 into T3, the form that binds directly to thyroid hormone receptors and switches on UCP1 gene expression, essentially priming the cell's thermogenic engine from the inside. Most of the detailed mechanistic work establishing this pathway, including studies on DIO2 deficient mice, comes from rodent research, and the coordination between thyroid signaling and adrenergic signaling appears tightly linked in those animal models. Each seems to amplify the other's effect on UCP1. When researchers have tried to confirm this picture in humans using PT-CT imaging, though, the results have been genuinely inconsistent rather than a clean replication. Maria Lahesma and colleagues, publishing in the Journal of Clinical Endocrinology and Metabolism in 2014, found that people with hyperthyroidism showed measurably higher brown fat metabolic activity than people with normal thyroid function. A separate study led by Avi Broders, published in PLOS One in 2016, followed thyroid cancer patients through a hypothyroid state and then a subclinical hyperthyroid state, and found a similar pattern. Higher thyroid hormone levels lined up with higher brown fat activity on their scans. Not every study agrees, though, and the disagreement itself is worth knowing about. Qingzhang and colleagues, publishing in Diabetes, Metabolism Research and Reviews in 2014, studied patients with Graves' disease, a condition causing severe hyperthyroidism, and found essentially no active brown fat in any of them, despite thyroid hormone levels far higher than in the other studies. Their proposed explanation was that skeletal muscle, which also ramps up its own heat production under high thyroid hormone, may have been competing for the same metabolic resources and masking brown fat's contribution on the scan. So the honest state of the science here is a well-established cellular mechanism sitting alongside human clinical data that hasn't fully agreed on how that mechanism plays out at the whole body level. Number 8. Sweat, not shiver, the muscle hormone that can trigger fat-burning fat. If thyroid hormone activates thermogenic fat from inside the bloodstream, this next trigger comes from an even more unexpected source: contracting muscle tissue itself. Pontus Bostrom and colleagues, working in a study published in Nature in 2012, identified a hormone released by exercising muscle that they named irisin, produced from a precursor protein called FNDC5 through a pathway dependent on the same PGC1 alpha signaling molecule involved in general exercise adaptation. In their experiments, irisin triggered white fat tissue to take on browning characteristics, increasing UCP1 expression and thermogenic gene activity. This finding generated enormous excitement, and it also generated real scientific pushback, which is worth being upfront about. The original discovery was made largely in mice, and when researchers examined the human version of the FNDC5 gene, they found a mutation in the start codon, the genetic signal that tells cells where to begin building a protein, that isn't present in mice or most other primates. This led one research group, publishing in Scientific Reports in 2015, to argue in a paper bluntly titled Irisin, a myth rather than an exercise-inducible myokine, that the human version of this pathway might not function the same way at all, or might not produce meaningful circulating iris in the way rodent muscle does. The picture isn't entirely settled in irisin's favor or against it. Frode Norheim and colleagues, publishing in the FEBS journal in 2014, put this to a direct test in humans. 26 previously inactive men completed 12 weeks of combined endurance and strength training, with muscle biopsies taken before and after. Both the PGC1 alpha gene and the FNDC5 gene showed measurable increases in muscle tissue following the training period, in both healthy participants and those with early signs of insulin resistance. What remains genuinely unresolved is whether that muscle level increase reliably translates into enough circulating irisin, doing enough biological work on human fat tissue to matter. A question different research groups are still actively arguing about, which is a reasonably honest place to leave it. Chili peppers get their heat from a compound called capsaicin, and the reason your mouth reacts to it the way it does comes down to a single receptor, TRPV1, a protein normally built to detect actual heat. Capsaicin essentially tricks this receptor into firing as though your tongue were touching something hot. What's less widely known is that this same receptor sits on sympathetic nerve fibers throughout the body, including the ones that run into thermogenic fat tissue, meaning capsaicin and cold exposure end up flipping a very similar biological switch, just through different doors. Takeshi Yonashiro and colleagues tested this directly in a human study published in the American Journal of Clinical Nutrition in 2012. 18 healthy young men underwent PT scanning after cold exposure to identify who had detectable thermogenic fat and who didn't, splitting the group into BAT-positive and BAT-negative categories. Both groups then ingested a concentrated non-pungent capsaicin extract. The result was telling. Energy expenditure rose meaningfully in the men who already had active thermogenic fat, and essentially not at all in those who didn't. Capsaicin wasn't creating this fat out of nowhere. It was only capable of switching on tissue that was already there. Separate laboratory work by researchers, including Kita and colleagues, using isolated brown fat cells rather than whole body studies, found that capsaicin can act on these cells fairly directly, encouraging the kind of gene activity associated with heat production even outside the context of the full nervous system pathway. Between the whole body human data and the cellular evidence, capsaicin looks like a genuine, if modest, activator sitting in the same family as cold and thyroid signaling. One more entry point into a system that, as it turns out, has several different doors leading to the same room. All of this biology would be an interesting footnote if it had no bearing on actual health outcomes. So here's where the picture gets more consequential. Tobias Becker and colleagues, publishing in Nature Medicine in 2021, ran one of the largest analyses of its kind. Over 134,000 PET-CT scans from more than 52,000 patients, sorted by whether or not detectable thermogenic fat showed up on the scan. People with detectable brown fat had lower rates of type 2 diabetes, unhealthy cholesterol patterns, coronary artery disease, and high blood pressure, even after the researchers statistically matched patients for age, sex, and body weight. This wasn't an isolated finding. A separate cohort study out of China, examining just over 4,000 adults, found a similar pattern. People with detectable thermogenic fat carried less visceral fat around their organs, had better fasting glucose numbers, and showed a healthier lipid profile than those without it. Two very different patient populations, scanned for entirely different medical reasons, converging on the same broad association between this fat type and a more favorable metabolic picture. But association is exactly the right word here, and it's worth sitting with that distinction rather than skating past it. These are observational data sets. People weren't randomly assigned to have more or less thermogenic fat. So it's entirely possible that some third factor, or even reverse causation, explains part of what researchers are seeing. The scientists behind these very findings have said as much themselves, which sets up a genuinely open question in this field. Does this fat type actively protect metabolic health, or is its presence simply a marker of a body that was already in better metabolic shape to begin with? That open question connects to a deeper problem researchers have only recently started addressing directly. André Carpentier and Denis Blondin, writing in the Journal of Clinical Investigation in 2023, made a case that complicates almost everything covered so far. The PET scans used in nearly all human brown fat research measure glucose uptake, not actual heat production. A tissue can pull in glucose on a scan without necessarily generating much heat at all, which means researchers may have spent over a decade using a proxy measurement that doesn't cleanly track the thing they actually care about. There's also a scale problem that rarely makes it into headlines. Rodents carry a much larger proportion of thermogenic fat relative to their total body size than humans do, and the dramatic metabolic effects seen in mouse studies, where activating this tissue can noticeably shift body weight and blood sugar, don't translate cleanly to human physiology, where the tissue makes up a far smaller fraction of total mass. Carpentier and Blondin argue explicitly that, unlike in rodents, human thermogenic fat on its own is unlikely to be a powerful enough lever to meaningfully treat obesity or diabetes. So where does that leave things? Researchers are now hunting for the genetic variants that might explain why some people carry more of this fat than others in the first place. and testing whether any behavioral or pharmacological strategy to activate it produces benefits large enough to matter clinically, not just detectable on a scan. Neither question has a settled answer yet. What started as a tissue written off as irrelevant in adults turned out to be real, active, and tied to genuine health patterns. But exactly how much it does and for whom is still being worked out in real time, one scan and one study at a time. While researchers keep working that out, the ordinary stuff, sleep, movement, steady meals, is still doing real, measurable work in the background. If you want a simple way to actually put that into practice, it's linked in the first pinned comment. If you made it this far, you clearly care about how your own biology actually works rather than the oversimplified version. So do me a favor and hit like, subscribe if you're not already, and send this to one person who still thinks fat only does one thing.